So this is my attempt at a mind map of a sort of a recap. So what we're going to do is we're going to start here, and we're going to be following the red arrows all the way through this information until we get to some sort of mental conclusion. Now, I'm going to be taking these gray detours every chance I get. All right? So follow along with me. So let's start here to here. With very few exceptions, all cells that make a multicellular organism share the same genetic material, the same genetics instructions, because they divide from the same progenitor cells via many repetitions of the cell cycle. The cell cycle results in two genetically identical daughter cells from one parent cell. And DNA is identical from generation to generation as a result of semi-conservative replication and addition to mitosis and cellular division. Now, we're going to go back to here, to the next part. So, in order for multicellular organisms to survive different tissues, different tissues need to perform different biological tasks. This is a division of labor that's called tissue specialization. So, one of the tissues that is specialized, a specialized tissue type, is the immune system. That's the immune tissue. It's tasked with the protection of the multicellular organism from outside invaders. Part of doing its job is the ability to recognize self from non-self. Self from non-self. Now, antigens on the surface of every cell in the body, and also outside the body in addition, help you to distinguish self from non-self. All cells will produce antigens based on instructions from their DNA. When you find non-self, when the immune system finds non-self, it is destroyed with extreme prejudice. All right, back to this. So down here is our next step. Stem cells are unspecialized cells that divide to form more stem cells and a differentiated cell that results in specialized tissues that vary in form and function, and and that form and function depend on their location and what tasks they need to perform. Detour. So once a cell has differentiated from a stem cell into a specialized tissue, it and all of the descendants of that differentiated cell will lose the ability to perform the tasks of cells of a different tissue type, even though all cells, as we discussed previously, have the same genetic material, the same DNA. So, even though they have the same DNA, once they differentiate, they can't perform the same tasks. Okay, back to here. Now, in order to perform tasks, a cell has to translate proteins from RNA, which RNA is transcribed from the genetic instructions of the DNA. A single strand of DNA, when you condense and coil that DNA, can be distinguished under a light microscope and is called a chromosome. So let's take a quick detour. All right. A segment or region of the DNA on a chromosome that codes for a specific protein is called a gene. Now, genes code for every aspect of a cell life, included when and how they will grow, divide, and die. So, every aspect of the cell can be controlled by turning off genes or turning them on. So if you make them able to be transcribed or not, that's turning them on or off. So you can control every aspect of the cell by controlling the genes. All right, let's get back over here. All right, moving down to the next one. DNA is contained within the nucleus of a eukaryotic cell, bound to histone proteins, and this becomes a 30 nanometer thick strand of coiled DNA and histones, which is called chromatin. Now, When DNA is tightly wound up in chromatin, the double helix cannot be separated, and neither can it be transcribed or translated. Neither can it be transcribed or replicated. So when you coil it, you can't transcribe it or replicate the DNA. Now, remembering again that in order to perform tasks, cells must translate proteins from RNA, which is transcribed from DNA. And also remembering that every aspect of the cell can be controlled by turning on or off genes. And also remembering that once a cell has differentiated, a stem cell differentiating into a specialized tissue will lose the ability to perform certain tasks that a tissue of another type could do, even though they have the same DNA. It's because you've turned on and off. It's because you've turned the genes on or off by making them, those genes being turned off means that you can't translate the RNA that would be made from that DNA. And that is how, and you can't translate the RNA because you can't transcribe it from DNA, because when it's coiled as chromatin, it can't be separated in order for transcription to occur at all. So that's how we turn on and off DNA. We make it such that we cannot uncoil and unwind and separate the DNA double helix. That way, we can't translate and we can't transcribe RNA and can't translate proteins. No proteins means no functions are performed. All right, so that was my mind map. All of this at once, starting here and going all the way through there, and all my little digressions. All of this leads back into the same idea that you can control the cell by turning genes on and off, and turning genes on and off means turning protein, making it so that you can't transcribe RNA, which can't be translated into proteins, and you can't do stuff without proteins. So that's everything all at once. Okay? Thank you. We're going to go back to our regular format now.